So good evening, everybody, and today we are going to start with our botany's first lecture, which is based on cell and its basic components and how does cell exist, how did cell exist, and what are the components of cell. And today we are going to discuss about cell wall. First, let us discuss what is cell. Cell is the basic structure and functional unit of life. As we know that according to the cell theory, the first postulate of cell theory, the cell is the basic structure and functional unit of life, and that forms the basic structure and function of the life. And then secondly, it is derived from pre-existing cell. Every cell is derived from the pre-existing cell, and that is why we are going to conclude this point that how life is derived from prokaryotes, which means we want to conclude here that eukaryotes are itself derived from simpler structures. Eukaryotes are having complex cellular structures, but these eukaryotes, according to the cell theory, are derived from pre-existing cells. As these are derived from pre-existing cells, these must have been derived from a simpler structure of their own. As they must have been derived from a simpler structure of their own, then simpler structure belongs to prokaryotes, as prokaryotes are having a simpler structure than eukaryotes. After which we are having another conclusion that life is derived from non-living things. That is quite bewildering as well as astonishing, amazing thing. But now I would be also providing you the scientific reason behind it as well. So as the cell theory is saying that it is derived from cell is derived from pre-existing cells. It is the basic structural function unit of life. It is derived from pre-existing cell through process of cell division, including meiosis and mitosis, which we will be discussing in our upcoming classes. And thirdly, there is a question: is life possible without cell? So we can say yes, it is possible without cell because there are acellular organisms. Acellular organisms such as virus, which do not have any cell. They are just containing a nucleic acid as well as they are containing a protein code with them. And whenever they go inside the host body, they become activated. They become alive. They become a living organism. So in that case, life is also possible without cell because virus is an acellular organism which is living in the host body. Now, when it comes to botany, botany is what? Botany is the study of plants and crops. What are plants here? Plants are individuals. Plants are individuals. After which we are having crops. Crops are what? Crops are a community of plants. What is for suppose I'm writing here as a community of plants. Botany is the study of plants and crops. Here plants is the word which is refers to the individual plants, whereas crops it refers to the community of plants, which means each crop is equals to one community of plants of a particular community of plants, such as wheat crop, rice crop belongs to a community of a particular wheat plants, rice plants. After which we are having two questions that life is derived from non-living things. So yes, life is derived from non-living things. How we are going to discuss it here. So before this, let us describe how eukaryotes are derived from prokaryotes. So uh, previously, scientists were not able to conclude that eukaryotes are derived from prokaryotes, but around 3.5 million years ago, 3.5 billion years ago, there was no existence of life. There was only the existence of prokaryotes, right? So those prokaryotes had a gene inside their DNA, not inside their DNA, inside their rRNA. They had a gene which is known as conserved gene. They had a gene known as conserved gene, and that conserved gene was having no mutations. It was having no mutation, which means it was unable to be evolved over thousands of centuries, which means it is unable to be evolved around thousands of centuries. If a conserved gene is present, it is unable to undergo mutation. It will not get mutated, so it would be evolved throughout centuries. It cannot be evolved throughout centuries. It cannot be evolved throughout centuries. Not evolved throughout centuries, and that is why the scientists today utilize that the gene for prokaryotes around 3.5 billion years ago, and that gene was compared with the genetic roots of eukaryotic cells gene. And so in eukaryotic cells, we are having rRNA as well, in which there is a conserved gene. But that is found where? That is found inside the mitochondria and chloroplast. In mitochondria and chloroplast, we know that these are autonomous organelles. These are autonomous organelles which contain their own DNA and RNA. As they contain their own DNA and RNA, they contain rRNA as well. They contain rRNA, due to which that rRNA contains the conserved gene, and that conserved gene is unable to mutate and evolve throughout centuries, due to which we can say that that conserved gene inside the eukaryotes was compared with the conserved gene inside the prokaryotes, due to which both of these concluded that yes, eukaryotes are derived from prokaryotes. Similarly, that is also concluding, that is also complying with the point of cell theory, that it is cells are divided from pre-existing cells. On the second or third part, we are here that life is derived from non-living things. Life is derived from non-living things, yes, because the prokaryotes, if eukaryotes are derived from prokaryotes, then how prokaryotes are divided, how prokaryotes came into existence. Prokaryotes are derived from what? Because prokaryotes are the simplest structure known. They do not have any other simpler structure than prokaryotes. And within prokaryotes, these prokaryotes were first derived from non-living things. These were derived from molecules. These were derived from molecules, and which constitute life. These are derived from biomolecules. These are derived from biomolecules which constitute life. That is why these are known as biomolecules in action, because these constitute life. So these biomolecules constituted life, and that was how life was derived from non-living things. And I hope that sounds clear. After which we are going to discuss about cell wall. What is cell wall? Cell wall is present in plants, algae, bacteria, and fungi. So what is cell wall? Cell wall is a non-living. It is non-living. It is dead. Also, there are some parts of cell wall which are alive, but generally we consider it as non-living. So after which we are having permeable. That it is permeable, completely permeable. Second, thirdly, it is protective, and fourthly, provides shape and support. And fifth, it is an outer layer. It is the outermost layer of the plant cell, algae cell, and bacterial cell and fungal cell. So it is present in plant cell, algae cell, bacterial cell, and fungal cell. In plant cell, it is made up of cellulose, hemicellulose, pectin, inorganic salts, and vexes. Whereas in the algae cell, it is made up of cellulose, melanin, and galactonin. If I'm not wrong, it's glucanin. Sorry, it's cellulose, menin, and glucanin. So algae is made up of cellulose, mannan, and glucanate. After which we are having the bacterial cell. Bacterial cell is made up of peptidoglycan, and fungal cell. Fungal cell is made up of chitin. What is chitin? Chitin is a polymer of NAG and NAM. What is NAG and NAM? NAG and NAM are two polysaccharides. NAG stands for N-acetyl glucosamine, whereas NAM stands for N-acetyl murmuric acid. These two combine together in order to form the chitin. Afterwards, we are having cellulose. What is cellulose? Cellulose is an insoluble polymer of sugar, which is insoluble in water. Due to which, whenever we have seen that whenever we pour water over the plants from upside, so these plants do not get swollen up, or the plant cells do not get flooded with water at the given moment. That is because of two reasons. First is because of the waxy cuticle, and secondly because of the cell wall. Cell wall contains the cellulose. Cellulose is insoluble polymer of sugar, due to which it resists the water to be entered inside the cell, due to which it doesn't allow water to go inside the cell, as it is itself insoluble in water, due to which it doesn't allow excessive water to enter inside the cell. Right afterwards, as it does not allow excessive water to enter inside the cell. We have seen that whenever we put water over the plant doesn't get flooded, doesn't get swollen. The plants do not get swollen because of the presence of cell wall. After which we have peptidoglycan. Peptidoglycan is part of a bacterial cell wall, which is um, made up of peptide. This is a conjugated molecule made up of peptide chain as well as glucose or carbohydrates. After which we are having glucanon and mannan. Glucanon and mannan are basically polysaccharides. These are polysaccharides which are made up of different structures, and we will be discussing that sometimes in another video. And here in components, we are having four components, which is primary cell wall, secondary cell wall, middle lamella, and tertiary cell wall. In among these four components, the tertiary cell wall is rarely found in plants. Rarely found in plants, although it is found in other organisms such as bacteria, algae, as well as fungi. So it is found in other organisms, but in plant cell wall, tertiary cell wall is rarely found. After which we are having the components of the cell. There are three components of the cell which we will be discussing here: middle lamella, primary cell wall, and secondary cell wall. So here I would like to present this diagram here. In this diagram, we can see that the first layer present here is known as middle lamella. What is middle lamella? Middle lamella is the cementing layer between two adjacent daughter cells. Cementing layer between two adjacent daughter cells, which is made up of calcium salts, magnesium salts, as well as pectin. And pectin is what? Pectin is a polymer of galacturonic acid molecules. And how many galacturonic acid molecules are combined together to form one pectin? There are 200 galacturonic acid molecules which combine together to form one pectin. And majority of the carboxylic groups which are present inside the pectin are methylated, which means the carboxylic group which are present inside the pectin are methylated. They contain COOCH3, as this is a formula of a methylated carboxylic group. Afterwards, that is hydrophilic in nature, which means it is hydrophilic in nature, but that is not as much hydrophilic as the pectic acid is, because pectic acid is more hydrophilic than pectin, whereas pectin is less hydrophilic. On the other hand side, that is a major component of middle lamella, but it is also found in the primary cell wall, which we will be discussing in the next part. So here, middle lamella is basically a cementing layer between two adjacent daughter cells, as well as it provides it is the outermost layer, and that's actually that actually forms a layer between that actually forms between the layer between the adjacent daughter cells. After which it is formed during cytokinesis. It is formed during cytokinesis. It's formed during cytokinesis. Let me write it here as well. It is formed during cytokinesis. It is formed during cytokinesis. And another interesting point, that the whenever we are having an unripened fruit, this unripened fruit is having middle lamella as a cementing layer in between is having a cementing layer in between the cells of that fruit, which is known as a middle lamella. Middle lamella acts as a cementing layer between the fruits of those between the cells of that fruit, due to which whenever the fruit starts to get ripened, the cementing layer which is present in between the two particular adjacent cells of that particular ripened fruit will start to vanish, will start to disappear, due to which the fruit starts to ripen. As the fruit starts to ripen, that causes to uh, generate and produce fully ripened fruit, and that generated fully ripened fruit is edible and can be consumed as a source of nutrition. So that is how it works. And middle lamella is basically sticky. How can we say that it is a cementing layer? So we can say that it is a cementing layer because it is having a sticky substance. It is having a sticky substance which is known as a gel of calcium pectate and magnesium pectate. Whenever the calcium and magnesium salts combine together with pectic acid, they form calcium pectate and magnesium pectate. Although pectic acid is hydrophilic, but whenever it forms calcium pectate and magnesium pectate, it becomes insoluble gel. It becomes insoluble gel. Insoluble gel. Afterwards, this is all about middle lamella. After which we are having primary cell wall. Primary cell wall is present between the middle lamella and the plasma membrane if the secondary cell wall is not present in the particular cell. Primary cell wall is most of the time present inside the mesosomatic cells and parenchymatic cells. This is present in mesosomatic cells and parenchymatic cells. Parenchymatic cells are cells. These are simple. These are cells of parenchyma tissues, and parenchyma tissues are themselves simple tissues, which are made up of only one type of cell. Mesosomatic cells are those cells which are young dividing cells. These are young dividing cells, which are actually able to and capable of growth. After which, this is a very thin layer. This is the thinnest layer because it is single layered. It is single layered as it is having a thinnest layer. And after that, we are having calcium, magnesium inside it. We are having pectin, cellulose, what are we having inside the primary cell wall? We are having the cellulose, pectin, hemicellulose, and pectin, cellulose, hemicellulose, and we are having, last but not the least, inorganic salts, but these are less found in primary cell wall, which means these are found in primary cell wall, but these are found really less. I mean, that is negligible amount. So how does the structure look like? So here, as you can see that here in the diagram, it's been given the structure of. Let me just zoom it a bit. Here it is given the structure of pectin, the structure of primary cell wall, which is having pectin, cellulose, microfibril, and hemicellulose. Now here, what we are having is that we are having uh, cellulose microfibrils. What are cellulose microfibrils? For understanding, for the structure of primary cell wall, here you can see that there are cellulose microfibrils. These are cellulose microfibrils. A cellulose microfibril is made up of three chains of cellulose which are bonded together. I guess let's just start it again. So cellulose microfibril is a set of three chains of cellulose which are bonded together with the help of pectin. So these three chains of cellulose which are bound together with the help of pectin is known as a single cellulose microfibril. A single cellulose microfibril is a set of three cellulose chains which are bonded together by pectin. Now this forms a single cellulose microfibril. Now the cellulose microfibril, which is here, as you can see, it is present over here. The cellulose microfibril in the yellow color is present over here. This cellulose microfibril is connected with another cellulose microfibril with the help of another connection with the help of another compound biomolecule known as the hemicellulose. Hemicellulose is made up of another compound known as hemicellulose. What is hemicellulose? Hemicellulose is again a polysaccharide and is made up of the following particles, which includes the manoglycans, diloglycans, mixed linkage beta glucans, which are supposed to be discussed in it is discussed in fields such as MSc and BSc. After which we are having what? After which that forms a basically particular structure of primary cell wall. After primary cell wall, what do we have is that after primary cell wall, that primary cell wall is having pectin in actual. So that pectin is actually made up of what? That pectin is actually synthesized by the Golgi body. Pectin is synthesized by Golgi body. You can understand it as I am not good at drawing. On the other hand side, we are having. So here, some MCQs point regarding primary cell wall here can be that for suppose, let me just adjust it a bit here. Yeah. So now the primary cell wall is made up of primary cell wall is made up of what? Primary cell wall is made up of the following components which we discussed here, known as hemicellulose, cellulose, pectin, as well as some inorganic salts. But primary cell wall is synthesized by protoplasm. It is an important MCQ point. After which we are having pectin. Pectin is synthesized by Golgi body. Pectin is synthesized by Golgi body. After which we are having another MCQ point that primary cell wall uh, is found in is found lifelong. These are found lifelong throughout lifetime. These have the capability to grow. These are found lifelong. What is mean by lifelong here? Lifelong means that these have the capability to grow. These have the capability of growth throughout the lifetime in growing cells. They have the capability of growth throughout the lifetime in growing cells. So suppose we have a pre-existing cell, pre-existing meristematic cell. That's meristematic cell which is actually growing in that condition. So suppose this is a meristematic cell. Now after a few months, after a few weeks, that turns into a larger meristematic cell by its growth. So this time what will happen? The meristematic cell will grow, alongside which the primary cell which is present inside the meristematic cell will also grow. And how does it grow? It grows by means of it grows by means of what? It grows uh, by stretching plastically. What does plastically means? It means it grows by stretching irreversibly. Moreover, what actually will happen here is that it will stretch plastically irreversibly. And how is it possible? It is possible because of the elastic nature. It is possible because of the elastic nature of primary cell wall. So it is having a plastic nature due to which it can stretch plastically so that it may grow alongside the meristematic cell which is grown. On the other hand side, let us discuss about our second part, which is the secondary cell wall. Here, secondary cell wall is present between the plasma membrane. Is present between the plasma membrane and primary cell wall. Is present between plasma membrane and primary cell wall. What is it composed of? It is composed of cellulose, lignin. It is composed of cellulose, lignin, waxes, inorganic salts, cellulose, lignin, waxes, inorganic salts, Sorry for the disturbance here. It is cellulose, lignin, waxes, inorganic salts, as well as due to a few deposition of lipids. I mean, Vexus is an alternative for lipids, which is present here. So that these are the components which constitute the secondary cell wall. After which, inside the secondary cell wall, we are having cellulose. The major component cellulose is present as a major component, which is, as you can see in the diagram, the cellulose. I guess it's not visible. It was visible in the previous one diagram. Here, as you can see, that there is cellulose cell wall is having a major component of it as the cellulose microfibrils. So here in the same case we are having cellulose microfibrils in the secondary cell wall. In the secondary cell wall we are having its major components as cellulose microfibrils. Again, the cellulose microfibril is made up of what? The cellulose microfibril is made up of pectin as well as it is made up of cellulose. That cellulose microfibril this time, rather than being joined together by hemicellulose, will be joined together by lignin. And lignin is what? Lignin is a polysaccharide, and that is a very strong, sticky polysaccharide which causes to provide strength, as well as causes to provide strength, shape, and mechanical support to the cell in which it is present, and overall to the plant in which it is present as well. So it is made up of the following components: cellulose, lignin, waxes, inorganic salts, and pectin. After which, let's second, as it is a secondary cell wall, it promotes secondary growth. Secondary growth means growth of the width, growth of the width, growth of the width of the plant is done by the secondary cell wall. By the way, secondary cell wall is not capable of growth, not capable of growth, not capable of growth. After which we can say that as it is not capable of growth, it is found in dead tissues. It is found in dead tissues such as tissues including tissues including tissues including parent, sorry, tissues including sclerenchymatic cells. Here, sclerenchymatic cells are cells which are also known as the sclerids, also known as sclerids or stone cells. These sclerids or stone cells are found in the nuts which we eat. Over the outer covering, the outer outer covering of the nuts is covered by a coat, is covered by a thick and strong coat, which is made up of these stone cells. These stone cells have that thick stone. We have these thick stone cells over them. And how does these stone cells make that particular nut coat as very strong? That is all because of the presence of stone cells, and stone cells are themselves containing cell wall which is having lignin, which is having a secondary cell wall which is having lignin. And that is why it makes the coating of the seed very much vigorous as well as strong, due to which it can becomes uh, inedible. After which we are having a few MCQs point as well, such as MCQ point including uh, secondary cell wall. The secondary cell wall. It's secondary cell wall. Unable to type it as secondary, so I'm writing it as second cell wall. I mean, you can understand it. Secondary cell wall is also known as lignified wall. Why is it not a lignified wall? Because it is made up of lignin. And primary wall is also known as the pectinized cell wall. It is also known as a pectinized cell wall because it is made up of pectin as a major component. After which we are having middle lamella. Middle lamella is also considered as the middle lamella is also considered as a pectinized cell wall. Why is it considered as a pectinized cell wall? Because it is having a pectin as a major component as well. So these two are considered as the pectinized cell walls, whereas the secondary cell wall is considered as a lignified cell wall. After which the primary cell wall is responsible for primary growth by primary cell wall. After which we are having middle lamella. What is the function of middle lamella? It is for cementing the material, cementing the two adjacent daughter cells, and this is formed during cytokinesis. During cytokinesis, phragmoplast occurs. During phragmoplast, what will happen? The formation of middle lamella will take place after the nucleokinesis. We are having karyokinesis or nucleokinesis. We are having the cytokinesis. The cytokinesis has a step in plant cells known as the phragmoplast. In phragmoplast, what will happen? The formation of middle lamella will take place. How it will take place? It will take place by the deposition of calcium and magnesium salts alongside pectin between the single parent cell. As it will become depositing between the single parent cell, it will cause to form a middle lamella. As it will start depositing the calcium magnesium salts as well as pectin, it will start forming middle lamella, which will be dividing that particular single parent cell into two daughter cells, and these two daughter cells will be adjacent to each other. And again, these will be connected with the help of middle lamella. That is why the middle lamella is formed during cytokinesis. After which we are having we are left with inside the structure with a few components on the first page, which are present, I guess, here. Yes. So here inside that we are having the lignin molecules. These lignin molecules are made up of types of lignin. Here we are having G lignin, S lignin, and H lignin. So G lignin stands for Guaiacil lignin, whereas S lignin stands for syringe lignin, whereas H lignin stands for rho hydroxyphenyl lignin. Rho hydroxyphenyl lignin. These are some names which are quite difficult to pronounce, but These are some parts. GSH lignins, which form the lignin of the secondary cell wall, as well as we are having lignin inside xylem vessels and tracheids. We are having lignin inside xylem vessels and tracheids. And xylem vessels are present in angiospermic plants only, whereas tracheids are present in angiospermic, gymnospermic, and vesicular plants as well. And xylem vessels are a bit, xylem vessels are a bit bigger in diameter, whereas tracheids are a bit shorter in diameter. Xylem vessels are a bit longer in diameter and a bigger in diameter, whereas tracheids are a bit shorter in diameter. Xylem vessels are basically having uh, vessel elements, vessel elements, as well as these are having side by side deposition of lignin. These are having side by side deposition of lignin, which causes to strengthen them and provide them with the mechanical support. After which we are having end-to-end connections, end-to-end connections between the elements, end-to-end connection between the vessel elements, and within the end-to-end connection between the vessel elements, there are also pits present. Pits are present end-to-end pits are also present, as well as alongside the end-to-end pits, we are not having a particular wall or a completely blocked xylem vessel, because xylem vessel has a major function of what? The xylem has a major function of moving the water from roots to upper parts of the plant, such as stem and leaves. Here, so for that purpose, it requires a lumen. So inside the xylem vessel, we are having an empty lumen, and this lumen is having no wall. It's having no wall. Rather, it is having some perforations. It is having some perforations. These perforations allow the movement of water up towards the upper side. These perforations allow the movement of water towards the upper side because of what? These perforations allow the movement of water towards the upper side during ascent of sap. We will be discussing that in our last chapter. So during ascent of sap, that will allow perforations. These perforations will allow water to move upward. And after that, these are some key points about the vessel elements. After which we have tracheids. Tracheids are having tapered endings. These are having tapered endings, and these are having oblique transverse walls. These are also known as oblique transverse walls because the oblique transverse walls. The word oblique stands for obliques means a slighted, which means a bit slighted towards something, and transverse means horizontal. Transverse means horizontal. On the other side, wall means any sort of boundary or layer. So here, object transverse wall refers to the slighted wall which is horizontally slighted. A wall which is horizontally slighted is known as the object transverse walls. So suppose this is the structure of vessel trachea. This is the structure of xylem trachea. Sorry, it is the structure of xylem trachea, and it is horizontally oblique. It is horizontally oblique, which means it is horizontally slighted. It is horizontally a bit slighted, due to which it was supposed to be called as obliquely transverse walls as well. I hope this sounds clear. After which we are having another functionality here, which is known as plasmodesmata. What is plasmodesmata now? Plasmodesmata, it is a channel between two protein. It is a channel between two adjacent cells. It is a channel between two adjacent cells without deposition of any material. That this is a channel which exists between two adjacent cells without deposition of any material. That allows the transport of nutrients from one cell to another cell. And that is actually just like a pathway or channel which connects two cells in order to transport materials from one cell to another. And that is actually a channel and can be blocked in some extreme conditions. So suppose in cold conditions, whenever there is a winter season, or whenever there is too cold in the environment, so what will happen? The plasma, also the cell wall is non-living; it is dead generally, due to which there will be no impact on cell wall. But the cell membrane is having fluid inside it. It is basically a fluid mosaic membrane that is having fluid with some mosaic of proteins embedded inside it. So that would be having what? That would be having fluid which can of course freeze in terms of cold weather, and as it will be freezing in a cold weather, what will happen? The fluid will be converted into ice crystals. Fluid will be converted into ice crystals, due to which these ice crystals can cause blockage of the channel, can cause blockage of the plasmodesmata, as it will cause blockage of plasmodesmata, as it will cause the blockage of plasmodesmata, it can cause a blockage of plasmodesmata, due to which what will happen? This can not allow the transport of nutrients from one side to another. For this, plants have another adaptability which we will study in our further topics, as it is the first topic. We will be keeping it quite simple. Plants have another adaptability for this. After which we are having what? We are having phragmoplast. I guess I have discussed about phragmoplast previously within this video, so I don't think I need to discuss it once again. As phragmoplast is a process during cytokinesis in which the formation of middle lamella occurs in order to divide the single parent cell inside into two independent adjacent daughter cells, which would be formed by formation of middle lamella due to deposition of calcium, magnesium salts, and pectin. And now let us briefly discuss about the functions of cell wall, which is to provide skeletal framework. Cell wall provides skeletal framework to plants. Also, as we know that the leaves are arranged perpendicular to the stem of the plant, so these leaves are having what? Even though these are arranged perpendicular to the plant, these are unable. These are basically have arranged perpendicular to the plant, due to which they might have just fall down over the ground. They should have fall down over the ground. They should have get teared up whenever there's extreme wind condition. But these do not do that. Why is that so? That is because the midrib of leaves is made up of parenchyma cells. It is made up of parenchymatic cells. These parenchymatic cells are having what? These parenchymatic cells are having excessive deposition of lignin. These are having excessive deposition of lignin and cellulose on the corners of the cell wall. It is having excessive deposition of lignin and cellulose on the corners of cell wall. As it is having excessive deposition of lignin and cellulose on the corner of cell wall, that can allow the cell to maintain its shape, as well as it can allow the cell to maintain its shape whenever there is an extreme wind condition, whenever there is water coming over it, whenever there is extreme wind condition, that can provide skeletal framework to it, that can provide shape, that can provide skeletal framework, so that the cells may not get teared up, and that the cells may not get teared up or get fell down on the ground, or not, or may not get any sort of damage at the moment. So this is all because of the cell wall, which contains the excessive deposition of lignin and cellulose on the corners of the polyclimatic cell wall, on the corners of polyclimatic cell wall. After which, another points include protection. It provides protection in two ways. First, it secures the inner constituents of cell. The inner constituents of cells, such as the cell organelles and nucleus, are protected. Secondly, it protects as a barrier. It acts as a barrier against pathogens, so that pathogens will not enter the cell. After which, we are having permeability. It allows the permeability of material, so that material is allowed to be moved inside the cell. So that the material is allowed to be moved inside the cell through diffusion. So the material is allowed to be moved inside the cell through diffusion, as well as it is also responsible for imbibition. Imbibition of water. How it is responsible for imbibition of water? That is actually hydrophilic in nature. As we previously discussed, that the cell wall is hydrophilic in nature, that is water loving. But some parts of cell wall, such as the cellulose, are insoluble in water, due to which it also prevents the excessive amount of water to be entered inside the cell. Due to which we can say that it also provides imbibition of water, but it also protects the cell from excessive amount of water which is provided to the cell. So it prevents excessive water. After which, it also provides shape, it provides support, as well as what else does it provide? It is actually having uh, support of two types, which is support of stem. Support of stem, it provides standing ability in stems, and secondly, it provides anchorage in roots. Anchorage in roots, and what is anchorage? Anchorage is the holding ability of roots. The roots of a plant are particularly hold it together. These are hold it together in a particular sort of bunch. That bunch is made with the help of cell wall, that causes the anchorage of roots, holding ability of roots, which is because of cell wall. These are some of the factors which I wanted to discuss here, and hopefully, I hope that it's all clear to you about the cell wall. We'll meet again with another new video. By then, that's all for today, and hopefully, we'll meet again. Thank you.